Good morning. Dr. B here with you again to finish up photosynthesis. We're going to talk about different means of producing ATP energy using pigment molecules such as chlorophylls in this particular video. In the first slide here, we're going to talk about production of ATP using the pigment chlorophyll with oxygen being absent in the environment. This particular process is called cyclic photophosphorylation. So you have a bacterium. It has thylakoid membranes in its cells. These thylakoid membranes contain chlorophyll molecules and also carotenoids. Light energy, photons of light energy can be absorbed by these pigments, and when they're transferred, when the energy is transferred to chlorophyll A, some of the electrons within the chlorophyll molecule can become excited, and they leave the chlorophyll molecule, where they are then passed into a set of membrane proteins where the electrons then lose their energy so they can return back to the chlorophyll molecule. When the electrons are passed through this set of proteins, they slowly release energy, and this energy that they release is actually used to form a chemical bond between ADP and an inorganic phosphate group to form ATP, the energy currency for cellular work. So, cyclic photophosphorylation assumes that a bacterium could have been a producer, just as glycolysis and maybe fermentation assume that a bacterium could have been a consumer, a heterotroph. This particular process of cyclic photophosphorylation is found in higher plants, eukaryotes. So this particular process has been passed on in the plant line of evolution. Excuse me. Oxygenic photosynthesis is also found in bacteria and is also found in higher plants. It involves two separate reactions called the light-dependent and light-independent reactions. For the sake of time, I'm going to move on and not really talk much about what is on the slide here, but I will talk about these bullets in the next few slides. So let's talk about the light-dependent reactions. So they occur within the thylakoid membranes. They involve clusters of several hundred pigment molecules that are called photosystems. There are at least two photosystems in plants. One is called photosystem 2, one is called photosystem 1. They absorb light energy a little bit different range of wavelength in the visible light spectrum, and that's where they get their names as photosystem 1 and photosystem 2. Well, when we get to the next slide, I'll mention that designation a little bit more. So photosystems absorb light, they absorb photons of light energy, and they transfer it to the primary pigment for photosynthesis, which is chlorophyll A. So chlorophyll A is where the light-dependent reactions will actually begin. As you see with the figure, it says chlorophyll B and carotenoids are called accessory. That means they absorb photons of light energy and they transfer it to chlorophyll A. So chlorophyll A is where the actual reaction of photosynthesis begins. It's called the light-dependent reactions. This figure shows you a Z-like configuration of the movement of electrons in what is called the light-dependent reactions of photosynthesis. So you see a phospholipid bilayer, that's the thylakoid membrane. In light green, you see two photosystems. photosystem 2 and photosystem 1. Photosystem 2 absorbs light the best when it has a wavelength of 680 nanometers. That's kind of in the orange-red range. Photosystem 1, you might see within that green square that it has P700. So light energy that it absorbs is best when it has a wavelength of 700 nanometers. So photosystems are clusters of several hundred different pigments with chlorophyll A kind of in the middle of this cluster of pigments, and that's where the reaction of the light reactions actually begins. The other pigments are called accessory that are in these photosystems. They absorb photons of light and then transfer that energy to chlorophyll A. So the light energy that's absorbed, again, excites electrons, just like slide number 2, cyclic photophosphorylation. Electrons are excited. When they're excited, they actually can leave the molecule, the chlorophyll A molecule, and they can pass through a set of membrane proteins, just like slide 2. They have passed through photosystem 2 to photosystem 1, and then they pass from photosystem 1 to an electron carrier that's called NADP, very similar to NAD that's used in respiration. The electrons that are removed from photosystem 2 are replaced by the splitting of water from the initial absorption of light energy. Hydrogen ions build up in the thylakoid lumen from the splitting of water, referred to as photolysis, and the hydrogen ions will be used in the production of ATP, as you see on the far right. So electrons are excited by the absorption of photons of light energy. The electrons pass through several sets of membrane proteins. We would call that an electron transport chain. The electrons are eventually accepted by an electron carrier molecule called NADP. The hydrogen ions build up in concentration to where they will eventually flow through an enzyme called ATP synthase that's in the thylakoid membrane. The energy that is released as those hydrogen ions move through the membrane will form a bond between ADP and inorganic phosphate to produce the energy currency ATP. Notice where ATP is produced in the stroma. Notice where NADPH is produced in the stroma. So the main function of the light reactions is to absorb photons of light energy. There are two photosystems that are involved. Light energy splits water, releases oxygen, releases electrons that are passed through two electron transport chains. The electrons and hydrogen atoms are eventually accepted by the electron carrier NADP. The movement of hydrogen ions results in the formation of ATP through the formation of a chemical bond. ATP and NADPH are formed in the stroma and are used in the Calvin cycle or the light-independent reactions, where carbon dioxide enters the cycle, and with 6 turns of that cycle, a glucose molecule can be produced. The Calvin cycle. In this particular example, 3 molecules of carbon dioxide enter at a time. They are bonded by the enzyme that's referred to as Rubisco. Rubisco is an enzyme that will bond carbon dioxide into this cycle. It forms a very unstable molecule that splits into phosphoglycerate. When this cycle turns 6 times, a glucose molecule can be released from this cycle. So in this figure, it says input 3 carbon dioxide, output a 3 carbon sugar. So if the cycle turns 6 times, then it's possible for a 6 carbon sugar to be released from this particular cycle. Melvin Calvin discovered and helped put together this particular cycle, put together what the reactants in the cycle were or are. He was awarded a Nobel Prize for putting this cycle together in 1961. You notice if this cycle turns 6 times, that there is 18 ATPs that are used, and there are 12 NADPHs that are used. So those ATP and NADPH molecules are formed in the light reactions. So they help to power this cycle and keep it running. So the Calvin cycle, or the light independent reactions, involve fixing carbon dioxide into a cyclical pathway, reducing the organic molecule through the addition of hydrogen, and then finally regenerating the molecule that will accept carbon dioxide into this cycle. With every 6 turns of this cycle, a 6 carbon carbohydrate can be released, and we typically call it glucose. Usually glucose is converted into sucrose for transport, table sugar, and then sucrose is converted into starch for storage once it is moved to where the plant is going to store it. We refer to plants that only use the Calvin cycle to fix carbon dioxide and produce glucose sugars. We refer to them as C3 plants. There are 2 modifications in the cycle of fixing carbon dioxide, and they are referred to as C4 plants. They are more efficient at producing sugars, and I really don't have time to talk about them all that much just for the sake of getting through this presentation. My suggestion is look at the videos and look at your textbook that goes over what C3 versus C4 plants are. This is the next to last slide in this particular video. Note what is required to produce a glucose molecule. Approximately 48 photons of light energy are absorbed. That splits 12 water molecules and releases 6 oxygen molecules. The production of ATP is typically 24 molecules. 6 tend to be a net gain. 18 ATPs tend to be used in the Calvin cycle or the dark reactions. The 12 hydrogen atoms that are released from water in the light reactions are bounded to NADP as NADPH. They are all used in the dark reactions or the Calvin cycle. And 6 carbon dioxides enter the Calvin cycle or dark reactions. And when that cycle turns 6 times, 1 glucose molecule can be formed. So that kind of summarizes how many molecules are required of CO2 and ATP and NADPH, how many photons of light energy have to be absorbed. Okay. And the final slide here is a chart that you could then use to study. On the left, you have the thylakoid membranes, and on the right, you have the dark reactions or the Calvin cycle. The light blue represents the stroma of a chloroplast. God bless.